Thank you so much for joining us today. Before we kick this off, I do want to point out to the audience that we welcome questions. Please utilize the Q&A section at the bottom of your screen. Type in questions as they come up, and we will address those at the conclusion. So I would like to now turn it over to Pierre, the CEO of Amyl. Please take it away, Pierre. The floor is yours.
Thank you, Kelly. I'm Pierre van der Papelier. Hello, everybody. Thank you for attending this seminar. I am the founder and CEO of Amyl Therapeutics, biotech startup based in Belgium. I am here with Damien Toulorge, who is our CSO and neuroscientist, expert in neuroscience. So I am happy to present you Amyl Therapeutics and our technology platform and our results, because we are convinced that we have really the treatment, the next generation treatment for neurodegenerative disease. To start, one question: who does not know someone affected by either Alzheimer disease or Parkinson disease? I think everybody knows someone or has heard of someone affected by those diseases. Those diseases are really devastating, not only for the patient, but also for the family, for the healthcare system. And it's a frequent disease. Worldwide, there are, in 2019, 55 million patients affected by those diseases. And the projection is that those numbers will increase to up to 140 million in the 2050s. Besides the despair of the patient and the families, there is also a huge cost for the healthcare system worldwide, and this cost will also increase. So we're talking about 50 million subjects affected by Alzheimer and 10 million by Parkinson, and there are other neurodegenerative diseases also. Neurodegenerative disease, you may know that amyloid fibrils, so those are amyloid disease, amyloid fibrils are involved in the pathogenesis, and they play a very important role, and especially 3 of them. And those 3 amyloid fibrils are represented at the bottom, amyloid beta, tau, and alpha-synuclein fibrils. And those fibrils come originally from the misfolding of proteins that start to aggregate, assemble as toxic oligomers, and then fibrils that then will go deposit in the brain or in the neurons, and then cause toxic effect and cause the death of the neurons and cause progressively the disease. And if you go back to the bottom picture, you see that, in fact, those fibrils have the same structure. As you can see, they are arranged the same way, and that's very important for our mode of action. Indeed, and the other thing is that those fibrils are involved in different neurodegenerative disease. AD on the top stands for Alzheimer's disease. A bit lower, you have PD standing for Parkinson's disease. And what you can guess from this cartoon is that, in fact, in Alzheimer's disease, there is always A beta, there is always tau, and in 50% of the case, there is alpha-synuclein also playing a role. In Parkinson's disease, there is always alpha-synuclein and tau. So it's not a disease of one amyloid fibril. It's those diseases are multiple, they are mixed pathologies. After decades of really failures, clinical failures and research failure in terms of treatment of those diseases, with no treatment being able to change the course of the disease. Now, since 2020, monoclonal antibodies targeting one of those 3 fibrils, amyloid beta, have demonstrated some efficacy and have been FDA approved. So the efficacy means that there is a 30% reduction in the progression of the disease. That's a very, it's a fantastic progress, and it's also a great hope for the current research and the current development. More recently, another monoclonal antibodies has shown some efficacy in phase 2, targeting anti-tau fibrils, which means that also targeting tau can have some efficacy. And in Parkinson disease, recent data also shown some efficacy by targeting alpha-synuclein, which means that by targeting those 3 amyloid fibrils, you can impact the course of the disease. However, we are not there yet. At the moment, as I said, only monoclonals against A beta, amyloid beta, are available, and they have a limited efficacy. So, in fact, to have a global impact on the disease, you would need to have different monoclonals against A beta, tau, and alpha-syn, and combination development could take decades and would certainly not reach the market before late in the 2040. The other thing is that the widespread adoption of those anti-amyloid beta monoclonal is limited by serious adverse events, the so-called ARIA. ARIA are brain edema and hemorrhage that are caused by those anti-amyloid beta monoclonals, and it happens in 25 to 30% of the patient. And therefore, for many physicians and patients, the risk-benefit ratio is not always seen as favorable in terms of efficacy. And the last thing is that, like for all monoclonal antibodies, there is only a limited part of the drug that is injected that pass what is called the blood-brain barrier that pass into the brain. So that's really a limitation of those therapies. What is the competitive advantage of Amyl Therapeutics? Well, a single product that can clear all type of amyloid from the brain. And that comes from, on the left part, you see a picture, and on the top of that picture, it's a binding site. And this binding site, called N1 and N2, will bind not to, like a monoclonal, to a very small part of the fibril, but will bind to the conformation of the amyloid fibril, because you remember and you see here, the conformation is always the same, which means that with one drug, one monoclonal, one fusion protein, we will bind and act on A beta, tau, and alpha-synuclein. It's like we had 3 monoclonals into one drug. And the bottom part of our protein is what we call a Fc domain. It's like in all monoclonal. It's there to attract the immune cells that will then, after binding to the amyloid fibril, it will then clean the brain from the fibril. So Amyl technology recapitulates the effect of 3 monoclonal in a single product. We have demonstrated many things, and we have plenty of results. As I said, in one, we have this universal amyloid fold binding. So with one drug, we bind to all 3. Well, after binding, there is attraction of the immune cells in yellow, number 2, and therefore cleaning of the amyloid fibrils, but also oligomers. On top of that, we have observed that not only there is a cleaning, a therapeutic effect, but there is also inhibition of the formation of the oligomers and of the fibrils, number 3. And therefore, there is a preventive effect. Some of those fibrils, like tau, are not outside the neurons, but are inside the neuronal cells. But they pass from one neuron to the next, and we have shown that we can block this passage. That's for the efficacy. For the safety, you remember that I mentioned the severe side effect called ARIA. we have modified, it's a proprietary modification, the Fc fragment to suppress the risk of ARIA. And finally, like several other companies, we have added what we call a brain shuttle that increased dramatically the passage from the blood into the brain. So which means that with a lower dose, we can have a higher concentration of our drug into the brain. Therefore, in conclusion, we demonstrated all desired property of a new breakthrough treatment for Alzheimer's disease, for Parkinson disease, and other neurodegenerative disease, with a unique efficacy with the removal of the 3-amyloid fibril plaques from the brain, stopping the formation of the 3-amyloid fibrils, and blocking the passage of the amyloid fibril from neuron to neuron. That's for the efficacy. It's also safe because we do not have the risk of ARIA. And we have the transport vehicle conjugation to enhance the brain delivery. We have numerous data to demonstrate that. It's not theory, it's reality. And we are now finalizing the lead optimization. In October this year, we'll have selected 4 preclinical candidates that will then further validate in different, for example, model of the disease in animals. Those are mice that are genetically modified to develop the Alzheimer's disease or Parkinson disease, which means that in October 26, we'll have selected our clinical candidate that will then be produced and for clinical development at the first clinical study, I will explain, will be in a specific population. The first clinical study will be in patients with Alzheimer's disease and Down syndrome, start in 2028 with preliminary results and clinical results in 2029. For that, to get to the clinical candidate, we are fundraising 5 million in equity. It's not much because in Belgium, in our region, in Walloon region, we are fortunate to receive generous grants, and we will receive 5 million euro in grants on top of the 5 million in equity. And then afterwards, to reach the clinical phase, we'll need to raise 15 million in equity, together with 15 million in grants from the Walloon region and from the European EIC. The current value of Amyl Therapeutics is quite low, as you can see, 15 million. But according to PwC, who made an estimation, an analysis, a full analysis actually, when we have the clinical candidate, that value will increase to 50 to 100 million euro. And when at the time of IND filing, it's going to be 500 million. And when we have clinical results, it will be in billions. It's not exaggerated because if you look at the deals nowadays, compound, for example, of amyloid beta alone, monoclonals targeting amyloid beta alone with a brain shuttle, that is without action on tau, without action on alpha-synuclein, without the safety modification, usually it's above the billion when it's reached the first clinical study in healthy volunteers. One word or one slide on the target population. Subject with Down syndrome, virtually all of them will develop Alzheimer's disease. It's an aggressive form of Alzheimer's disease with an extensive amyloid beta and tau pathology, with 10% of them showing also alpha-syn pathology. They develop that disease early in age, 35 to 40 years of age, and it contributes to the deterioration of their life, and they're also at higher risk of developing the cerebral hemorrhage, the so-called ARIA, so that the current immunotherapies cannot help them. It's a rare disease. We think that we can get an orphan drug designation, And the patients are readily available in their cohorts of patients in the US and in Europe. So that means that for clinical trials, we could have a relatively fast recruitment of patients. And finally, last but not least, to evaluate the efficacy, there are several biomarkers of neurodegeneration and of cleaning of the amyloid beta and tau that would help us for assessing efficacy. So this population, those subjects, they have no medical solution to help them. And at the same time, results that we will generate in those subjects will be translatable, applicable to the general Alzheimer's disease patient. We think that we are doing now what patients need to beat market expectation. As I said, in 2020s, the first monoclonal antibodies against amyloid beta have come to the market. In 2030, the first anti-tau and anti-alpha-synuclein monoclonals, individual monoclonals, will come on the market. And it will take another 5 years to have monoclonals without the side effect. And the real combination of the three monoclonals without ARIA and with brain shuttle will not come before in the 2040s. We are convinced that in our plans, we can be on the market in the indication of Down syndrome, Alzheimer's disease subject in 2032, and 5 years later in the so-called classical Alzheimer's disease. We have one product that can address disease caused by disorder caused by amyloid beta, tau, and alpha-synuclein. That's Alzheimer's disease and other type of dementia. But with the same technology, we can also address the Parkinson disease and related disorder where tau and alpha-synuclein are involved. And because there are also other type of disease that are caused by amyloid fibrils like light chain and TTR amyloidosis, we could also develop product for those diseases, which means that we do not have one product, but we have a pipeline of product. We are still a small group, very dedicated and motivated group here in Belgium. But we progress very fast because we collaborate with many groups, academic and private groups worldwide, in the US, in Europe, in China, because we are going with groups where the expertise is, and we do not want to lose time and develop everything in-house. No, what we are doing is we import the expertise when we have the results and the assay that are validated. The management team is small groups, is limited. I have a MD, PhD. I have worked in big pharma and in small biotech. I have the experience of product development and also as CEO. Damien Toulorge, our CSO, has the experience as neuroscientist, been trained in Paris, and also he has the experience of having developed himself in a biotech company. And then we have the support for financial and legal and IP. We have a strong IP portfolio that we fully own. The board is led by Florent Gros, co-founder. He is also a serial entrepreneur, patent attorney, venture partner. And we have as other member of the board, we have Hélène Sabatel for Nochac, a Belgian fund, Valérie Calenda from Merieux Equity Partner, a French fund, and Kenneth Bergfire, who is from New York. In summary, we have this unique mode of action acting on all amyloid fibrils in neurodegenerative disease. We have extensively characterized the mode of action. The technology and the method are fully validated. We have a strong and fully owned IP protection. We are raising 20 million euro Series A extension to generate clinical data, in parallel, we receive generous non-dilutive funding. And the use of proceeds of this 4-year plan, it's that we'll have multiple molecules against neurodegenerative disease and a clinical phase 1/2 proof of concept in Alzheimer's disease associated with Down syndrome. And I thank you for your attention, and we'll be happy to answer all questions you may have.
Great. Thank you so much, Pierre. Yeah, there's some questions that have come in, so let's get to those. And I'm going to have you stop sharing your screen. Actually, I can probably do that for you. Oh, perfect. Okay. So what is the company's long-term vision? Do you see Amyl focusing narrowly on Alzheimer's or broadly across multiple amyloid driven neurodegenerative conditions?
Our long-term vision is to develop products for all neurodegenerative disease wherein amyloid fibrils are involved, but also besides other amyloid-mediated disease like amyloidosis, for example.
Thank you. And then how do you prioritize which indications to pursue first, and what criteria drive those decisions? Scientific rationale, regulatory pathways, commercial opportunity, etc.
Sure. Our first indication will be Alzheimer's disease, and the very first indication will be patient with Down syndrome and Alzheimer's disease, because there is an important medical need, but also because we are a small company. It's a small population, orphan or not, but a small population. For the clinical development will be easier for us. The clinical development can be faster and easier than in a general Alzheimer's disease population. At the same time, the results that will generate in those subjects will be useful and applicable for the classical Alzheimer's disease patient. It's very likely that after we have clinical data, we can license out the product for one indication like Alzheimer's disease, so that it generates revenue to allow us to move forwards in other indication.
It sounds like your first target is DSAD. Why are you targeting this population versus AD or PD patients, which are a much larger population?
So we have selected that population because to us it's going to be not only we fulfill a medical need, an important medical need, but it's also a very good and easier model than targeting directly Alzheimer's disease or Parkinson disease. As we said, it's a severe form of the disease with the higher risk of brain side effects. If we demonstrate efficacy and safety in that population, it automatically will translate into results that can be used for the Alzheimer's disease population. The other thing is that the cost for a small company and to really develop now, especially that there are competitors on the market, to develop a drug for the general Alzheimer's disease will be out of reach for our company at our stage.
Thank you. Have you observed any improvements in cognitive or functional outcomes in animal models beyond biomarker reduction?
At this stage, we have not yet performed those animal studies. This is our target for the next year, and that's why we are raising the 5 million euro, is to generate data in efficacy disease animal model. So that is going to be mice that are genetically modified to develop amyloid beta Alzheimer's disease or tau Alzheimer's disease or Parkinson disease, for example. And so we will finalize the selection of our candidate in those animal models. All the plans are ready, and we are really ready to start those animal studies to have select the final candidate for clinical development in October 26.
Thank you. And given the competitive landscape in Alzheimer's and related diseases, do you see your company as an eventual partner to big pharma, or or a company that could build a full commercial organization independently? And what is your plan for the future, going public or sell to the pharma companies?
I think it's a mix of all of that. As I said, we are a small company. Eventually, when you have the first clinical data, given the contact we already have with big pharma, and many of them are following us very closely, they will be interested to acquire one of our products for a big indication like Alzheimer or Parkinson. That will give us sufficient financial means to pursue the development in other indication, and then eventually, of course, we will become public. But that's we will need to have clinical data, phase 2 data, and also a prospect in terms of results before we go public. You know that nowadays becoming public as a biotech, it's quite challenging.
Thank you. What preclinical evidence suggests the lower risk of ARIA?
I did not present that, but we have developed, and Damien could explain that in more details, but we have developed an assay. So ARIA are caused by the fact that in the brain of Alzheimer's disease, there are plaques of amyloid beta that are everywhere, but also around the blood vessels in the brain. And when the drug, the current monoclonals, will then attach to those amyloid fibrils, the plaque, the immune cells come and destroy this complex of the monoclonals and the amyloid fibril to clean the plaques. But it's an immune cell, so it also induces a local inflammation. This local inflammation causes edema. Since it's close to the blood vessels, it also causes hemorrhage. So the inflammation is a core mechanism of those side effects. What we have done is that we have made a culture of macroglia cells. That is, those are the inflammatory immune cells that are in the brain. And we have added the amyloid beta plaque. And then either the monoclonal that is on the market, anti-beta, lecanemab, or our product, after modification of the fragment of our product. And what we see is that we maintain the same level of what we call phagocytosis, that is the destruction of the amyloid fibril plaque, than lecanemab. But where lecanemab, there is a high secretion of the inflammatory cytokines, so the one that cause inflammation. With our product, we see more than very dramatic reduction of this amyloid, inflammatory cytokines. That means that we keep the same efficacy in terms of cleaning by phagocytosis by the immune cells, but without the inflammatory surrounding. And therefore, with this, we will decrease and attenuate greatly the side effect. There is another mechanism that can play a role. I mentioned the brain shuttle that facilitate the passage into the brain. The current monoclonals that are on the market do not have that brain shuttle. But Roche, the huge Swiss pharma group, presented in April this year data clinical data of a monoclonal anti-amyloid beta like the one that are on the market, but with a brain shuttle that facilitate the passage into the brain. And this addition of a brain shuttle also decrease the risk of side effect, which means that we have two mechanisms in our product that will decrease the risk of those brain side effects.
Thank you. What assays or imaging modalities are you using to track amyloid clearance and safety in your preclinical studies?
Well, I leave that to Damien, who will explain what we will
Yes, we have a lot.
measure.
In fact, we have measured a lot of things. We have measured the binding by at least 6 different methods: SPR binding, binding by dot blot, binding detected by electron microscopy. For the clearance of the plaque, we performed it in 2 different in vitro assays using either a human cell line of monocytes, cell line called THP1, or also with human microglial cells derived from iPSC. We are also doing all the binding experiments ex vivo. We have it in mouse models of A-beta pathology, of tau pathology, and we also very recently also demonstrated the binding to human patients' brain who was suffering from Alzheimer disease. So this is a very important milestone for us because it demonstrates that yes, we have the capability to bind to the aggregates directly inside the brain of Alzheimer disease patients. I'm obviously, and for the future, it will be mainly the in vivo proof of concept of efficacy that will be performed in mouse model of Alzheimer disease pathology, either amyloid beta or tau pathology or alpha-synuclein pathology. We will demonstrate it in all 3 different mouse models where we will evaluate the clearance by evaluating either for amyloid beta the surface occupied by the plaques, amyloid beta plaques, and for tau and for alpha-synuclein, it's by evaluating the levels of alpha-synuclein and tau pathology directly inside the brain by biochemical measurements. And all this will be completed with symptomatic effect, not symptomatic effect, but effect on symptoms, cognitive performance, and so on, which are characteristic in this in these models.
So no imaging, but we look directly in the brain.
Yeah.
Okay.
Thank you. Have you engaged in any early dialogue with regulators such as EMA or FDA about your development plan, and what feedback have you received?
Well, we have also worked, and currently it's on hold, but on amyloidosis, and we have engaged with the European authorities, and the feedback was quite positive. Our plans for this program in Alzheimer's disease is to meet with both European authorities and the FDA in Q2 2026. Yeah, yes, 26, correct. Yeah. So it's going to be in 9 months from now. That's the right moment in terms of the results that we can present, but also to agree with them on different toxicology and clinical plans.
Thank you. And the last question has come in about, your raise. Could you just reiterate how much you're looking for and what minimum investment size would you accept?
Well, we are raising 5 million to reach end of 2026. So 5 million euro. It's somewhat a bit more in the US dollar, but doesn't matter, because we also have confirmation that we will receive virtually the same in terms of grants, so that will allow us to reach end 26, early 27 without any problem. That's what we need. In terms of ticket size, the largest, the best, of course, but there is no specific limitation.
Okay, great. Thank you. Well, that concludes the questions. And for the audience, if you have additional questions or want to be connected to the team here, please reach out. Everyone has my email address. I'm happy to facilitate that. In turn, they will have all of your information as well, and we'll be reaching out. Pierre, I'm going to turn this back over to you for any final words before we close out.
Yes, so thank you for organizing this webinar and for attending it. We are really, as I said, we are really a small but very motivated and dedicated team. And this is the reason is that now we are all convinced that we have in hand a product that can help millions of people. And I can say personally, my father died of Parkinson's disease. And if I had that kind of product, even in early phase, I would have tried to help him, because I know how desperating that kind of disease is for the patient and also for the families all around. And here I am à la fois both enthusiastic because we have really great results that nobody else can claim, and at the same time I'm a bit desperate because of the funding. Funding of biotech is very difficult nowadays. The classical VCs funds are all saying, "Oh, we keep our money to develop all the companies that we already have in our portfolio, and we do not want to invest in new companies." And it would be a pity that we cannot develop this product just because of that well, economical and political situation worldwide.
Great. Thank you so much. And thank you everyone for joining us today. And I hope everyone has a great day.